Welcome, everyone, to the afternoon session. Our first speaker is Rachel Wilson from Harvard Medical School, and she will tell us about thermosensory processing in Drosophila.
So it's been known since the 19th century that peripheral neurons have specific sensitivities for warm or cool stimuli. This was shown by the work of several investigators, including Magnus Blix, pictured here. So he didn't use fancy optogenetic techniques. He just used a fine needle, which he inserted into his own skin. And he passed a small amount of electrical current through that needle. And he found that depending on where he placed this needle on his skin, here this is a map of his own wrist, he was able to elicit either a percept of warming or a percept of cooling. And we now know that he was, in effect, stimulating individual peripheral thermosensory neurons in the skin. So since then, I think the picture of how these kind of peripheral signals are processed in the central nervous system has not become particularly clear, to say the least. So there are several competing live theories about how this might work, just described in a recent review that I liked. One theory is the idea that essentially these different somatosensory neurons, for example, warm and cool peripheral neurons, essentially make independent contributions to our sensory percepts, such that, for example, the warm preferring neurons might contribute always and only to the percept of warming, and the cold preferring neurons might contribute always and only to the percept of cooling. And if you were to co-activate these neurons, you might get something like a weighted linear sum of their activities, giving rise to a particular percept. And this is sometimes called the labeled line model, although I think that's kind of a confusing name. There's a different idea out there, which is that neurons in the periphery, like warm and cool neurons, might actually form opponent pairs that mutually inhibit each other in the central nervous system. And then there are other kind of variants on the idea of central combinations. For example, this idea that's been called mixing or coloring, such that, for example, signals from one channel might give rise to both excitatory and inhibitory inputs to the other channel, and the net role of that channel might then be determined by the balance of excitation and inhibition. And so the reason that these are all kind of still live theories in 2014 is that there's some indirect evidence supporting each of them. So for example, the evidence for independent processing of warm and cool stimuli comes from experiments like this from David Anderson's lab. What they did here was to essentially ablate peripheral neurons that express TRPV1, which is a putative warm activated channel. This is a slice through the spinal cord showing that these neurons have been ablated. And the finding here is that ablating these warm sensitive fibers, warm activated fibers, diminishes behavioral avoidance of warm stimuli, but not avoidance of cool stimuli. However, there's also evidence in favor of non-independent processing of warm and cool stimuli, coming from, for example, psychophysical illusions like the thermal grill illusion. In this apparatus, these metal bars are alternatingly warmed and cooled. So you have warm, cool, warm, core, warm, core. And when you touch them individually, they feel innocuous. But if you place your hand simultaneously on this whole grill, you have a percept of intense burning hot pain. And that's clearly not sort of a weighted average of the percepts created by these individual components. It seems to be a synthetic emergent property of co-activating these afferents. So we decided to investigate how peripheral thermosensory inputs are combined in the CNS using the fly as a model system. One of the virtues of the fly is that we can perform in vivo whole cell patch clamp recordings in awake animals from genetically identified neurons in the brain. And a recent study showed that the fly has peripheral thermosensory neurons here in the antenna at the base of this feathery structure called the arista. And then importantly, these peripheral neurons are selectively excited by either warm or cool stimuli, just like warm or cool neurons in the human or mammalian periphery. And so this is the study I'm referring to. It was published by Charles Zucker's lab. And what I'm showing you is a false color rendering of 2-photon calcium imaging data from that paper. And what you should notice is that in this image of a cluster of neurons here at the base of the arista. Some of these neurons are selectively depolarized by warming, and others are selectively depolarized by cooling. Here's I should mention that the warm neurons express a gene which is going to be important for this talk, which is called GR28BD. For all intents and purposes, this seems to be the warm peripheral transduction molecule. It's required for warm-induced behaviors in the fly, and importantly, if you misexpress this receptor in neurons that normally are not thermosensitive, you confer the kind of extraordinary thermosensitivity on those neurons that you normally observe in the warm selective neurons. So the main thing is that this is probably the warm transduction molecule. Interesting, we actually don't know what the cool transduction molecule is yet. So here's more data from this really interesting paper showing that in response to a warm stimulus, the warm cells are depolarized, but importantly, this is going to be important for the talk, the cool cells have their activity pretty profoundly suppressed. So the conjecture is that there's a rather high level of basal activity in the transduction machinery, which is suppressed by the non-preferred stimulus. And conversely, cool cells are excited by cooling, but warm cells also have some basal level of activity, which is suppressed by their non-preferred stimuli. So these are not so much warm and cool neurons as thermometer neurons. One of them is just a right-side-up thermometer neuron, and the other one's an upside-down thermometer neuron. So the question we asked is, how does the brain process information from peripheral thermosensors? So the first order of business was to create a way to specifically manipulate the temperature at the antenna. So we're doing this by running a laminar stream across the antenna. It's just about 150 microns in diameter, so about the dimensions of the antenna itself. We can show when we record from neurons in the brain that all their thermal responses are abolished by cutting the antennal nerve, indicating that the stimulus is truly in the periphery. And this is an example of kind of the typical stimuli I'm going to show you responses to. So here we're rapidly cooling the periphery by about 10 degrees from room temperature. Here we're rapidly warming it. These rapid steps occur over about 100 milliseconds. But we can also cool or warm the periphery more slowly. So we can see the same jump from room temperature over a period of several seconds. So usefully, some rather old literature showed that peripheral thermosensory neurons actually project to a distinct brain region which has already been identified. So the old-fashioned technique is to sort of cut or nick this peripheral structure, the arista, dunk the antenna in a cobalt solution, and cobalt is taken up by the cut axons. And what you see is the axons of these peripheral thermosensory neurons selectively innervate this region that's just posterior to the antennal lobe. So the first thing we did was to visually screen driver lines, GAL4 lines, looking for neurons that had dendrites in this brain region, neurons that might be postsynaptic to peripheral thermosensory neurons. So the graduate student who did this work, Wendy Liu, visually screened about 7,000 lines from the public collection of the HHMI Genelia Farm Research Campus. And then she used interesting lines to drive GFP expression, and she performed in vivo whole cell current clamp recordings from these neurons in order to characterize their thermal responses. And she identified in total about 4 types of projection neurons in the brain region of interest. I'm going to call these projection neurons or principal neurons. These are neurons that by definition project to higher brain regions. You might also be interested in local interneurons, and I'll touch on those really briefly at the end. So I'm going to walk you through these 4 types of projection neurons. And as a spoiler, I want you to know that the first half of the story is, frankly, a little bit boring. But if you don't pay attention to it, you will probably be a little bit lost in the second half of the story, which at least, in my opinion, is much more interesting. So just hang with it for the next few minutes. All right, so the first type of projection neuron or principal neuron I'm going to tell you about are cool projection neurons type 1. And that should tip you off to the fact that there is a second type of cool projection neuron coming your way in a few minutes. So these neurons are depolarized by cooling, and they're hyperpolarized or inhibited by warming. You'll see that memory potential changes here are accompanied by an increase in firing rate. We go from a low rate of spontaneous spiking to quite a high rate of evoked spiking. In these neurons, as in most Drosophila central neurons, the spike initiation zone is a little distance from the cell body, meaning that spikes are slightly attenuated by cable filtering as they move toward the cell body, which is where we have our electrode. and that's why the spikes look a little bit short, but they are sodium spikes. Okay, and so you'll notice that this neuron is kind of robustly excited by rapid cooling and somewhat more modestly excited by slower cooling. Here are peri-stimulus time histograms, so I'm plotting firing rate as a function of time. You can see a large transient burst of spiking at the onset of a rapid cooling step, and then a lower evoked firing rate at the onset of a slower cooling step. So we think that the circuit that gives rise to these responses is actually pretty simple. So this is essentially what we think it looks like, and I'll tell you why. So these are, again, the normal responses to our rapid cooling and warming steps. If we bath apply pharmacological antagonists of synaptic inhibition, so just antagonists of GABA receptors essentially, we really have very little effect on the stimulus-evoked responses of these neurons, arguing that they're not powerfully modulated by inhibition. And also, if we repeat these experiments in the GR28BD mutant, you'll recall that this is the putative transduction channel for warming, transduction molecule at least. And so it ought to silence these warm peripheral neurons. There's, again, really no effect on the responses of these neurons. Perhaps that's not surprising. It looks, for all intents and purposes, as if they're excited by cool peripheral cells, and that's it. So we think the circuit for the central representation of cooling is pretty simple. These type 1 cool projection neurons actually project to a brain region called the lateral protocerebrum, which if you're a fly person will mean something, and if you're not a fly person, you won't care. I'll just say that it's a higher brain region involved in multimodal sensory integration and ultimately sensory motor transformations. Okay, so we also found a second type of cool projection neurons. These neurons are different in a few ways. Again, they have some basal level of spontaneous activity. They're depolarized by cooling. Interestingly, at some point during their depolarization, they often fire a burst of sort of complex spikes, just a hallmark of their unusual intrinsic properties. But you'll notice that these neurons differ from the type 1 neurons in a couple respects. First of all, their peak level of activity seems to depend mainly on absolute temperature rather than the rate of change of temperature. So they show similar evoked firing rates in response to a fast versus slow cooling step. Another thing you'll notice is that in contrast to the first type of neuron, the responses are relatively non-adapting. They're pretty tonic rather than phasic, again suggesting that at least on a time scale of a few seconds, they're encoding something close to absolute temperature rather than rate of change of temperature. And again, various kind of mechanistic manipulations designed to perturb potentially other kinds of more complicated inputs of this cell really had no effect. So when you block inhibition, not much happens. There's a modest disinhibition of the initial peak of the response. There's a little bit of disinhibition of spontaneous activity. Not much else happens. And again, the mutant in the putative warm transduction molecule has very little effect on the responses of these neurons. So again, we think that the circuit that gives rise to the representation of cooling in these neurons is also relatively simple, dominated by feed-forward excitation from cool peripheral cells. Interestingly, these type 2 neurons project to a distinct higher brain region called the mushroom body, which is quite clearly the locus of associative learning and memory in the fly. So it's kind of interesting to meditate on why this locus of learning and memory might receive information about absolute temperature, whereas the other brain region receives information about the rate of change of cooling. In any event, we then characterized a third cell type that I'm going to call warm projection neurons. So these neurons are excited by warming, as their name would imply. They show robust firing rate increases in response to rapid warming and more modest firing rate increases in response to slow warming. And these neurons, as we've seen before with other neurons, are inhibited. Their activity is suppressed by the non-preferred stimulus, by cooling. But one thing that's sort of interesting to note here is the fact that although these neurons are net hyperpolarized in response to a cooling stimulus, if you sort of look carefully at the memory potential, you'll see that the rate of excitatory postsynaptic potentials actually increases during this stimulus. And what that suggests is that although the net input to this neuron in response to a cool stimulus is inhibitory and/or a withdrawal of excitation, they seem to be getting excitatory input from some neurons that are excited by cooling. Interestingly, when we blocked synaptic inhibition in these recordings, we modestly disinhibit the heating-evoked excitation. But what we unmasked here is a cooling-evoked excitation. Again, consistent with the idea that these neurons get excitatory input from neurons that are excited by cooling, and that this input is normally masked perhaps by cooling-evoked inhibition, which operates in parallel. So that's led us to hypothesize that there might be essentially a more complicated circuit that gives rise to these representations of warming in the brain. So it's logical to think that these neurons get excitation from warm peripheral neurons. But the fact that we see an increase in EPSPs in response to a cool stimuli suggests that they might get perhaps a weak input from cool peripheral cells. And the finding that that input now becomes unmasked suggests that there might also be in parallel cool-evoked inhibition, which is going through some hypothetical interposed inhibitory interneuron, right? So we're going to try to put parts of this model to the test. First of all, this predicts that there actually are two ways that this neuron can be excited in response to warming. Peripheral warm neurons provide excitation to this neuron, but this hypothesis would suggest there's another pathway as well. Remember that the cool cells are profoundly inhibited by warming stimuli. If they drive spontaneous or tonic activity in these interposed interneurons, then what you'd see is a disinhibition in these warm cells, and that might give rise to some warm-evoked excitation as well. So what we find actually is consistent with that. So in this mutant, the GR28BD mutant, again, this is a mutant in the putative transduction molecule for warming in the periphery. We see that warming-evoked excitation is essentially intact, provided that the stimulus is a rapid warming. If the stimulus is a slow warming, we see a very substantial attenuation in the warm-evoked excitation. This would tend to suggest then that for a slow warming stimulus, excitation is driven primarily by the peripheral warm cells. But if the stimulus is sufficiently rapid, what you get is a really large disinhibition that drives a burst of activity in these cells. And consistent with that picture, if we look at the mutant and we just block synaptic inhibition with bath application of antagonists of inhibitory neurotransmitter receptors, we see this warming-evoked excitation is totally abolished. Now, for a neurophysiologist, that's not what you normally see. When you block inhibition, things typically go nutso. You see lots of excitatory responses. So this is very striking. And this is quite different from what happens in wild-type flies. Again, in wild-type flies, blocking inhibition does not block warming-evoked excitation. If anything, it disinhibits it. So this is compatible with the idea that this residual response in the mutant, where there are no warm peripheral cells, is being driven by disinhibition, and therefore it requires synaptic inhibition. So this is the idea that there's a direct pathway for warming-evoked excitation in these cells, but there's also an indirect pathway that can function when these cells are not present. It consists of a disinhibitory loop, but it's only efficiently recruited by a very rapid warming stimulus. So we wanted to put this circuit model to a more stringent test. We were kind of worried about the following caveat. I think there's good evidence that GR28BD is the peripheral transduction molecule for warming, but I would say that that evidence is sort of not as strong as you might ideally like. We wondered whether there might be, for example, a secondary peripheral warm sensor expressed by these neurons, such that they had some residual warm-evoked excitation in the mutant. We can't directly look at the warm-evoked responses of these cells in the mutant for a stupid technical reason that's plagued both our lab and the lab that initially reported this finding, but we wanted to test this idea in another way. We wanted to directly silence these cells rather than silencing this transduction molecule. So here we took advantage of a pharmacogenetic strategy we developed a little while ago. So the idea is to misexpress a histamine-gated chloride channel in the neurons that we want to silence. This molecule, ORT, is very nice. It's an endogenous Drosophila gene, but its function is mainly restricted to the first synapse of the visual system. It's a homomeric channel with a really large conductance, and it shows very little desensitization. So it's kind of an ideal effector for silencing neurons. So what we did in this kind of pilot or proof of principle experiment in the olfactory system is we misexpressed this channel in peripheral olfactory neurons, not thermosensory neurons, but olfactory neurons. And we know the wiring of the olfactory system very clearly, and so it's a nice proof of principle experiment. In this experiment, then, peripheral olfactory neurons express the channel. When we bath apply histamine, we really wipe out spontaneous activity, substantially attenuate odor-evoked depolarization. Here's group data, and here's the genetic control showing that in flies that are wild type, there's essentially no effect of histamine. So now we're going to use this technique to silence the peripheral warm activated neurons rather than peripheral olfactory neurons. And the result, I'll just tell you, essentially phenocopies the GR28BD mutant. So in the absence of histamine in this genetic background, we see pretty normal responses. When we silence the peripheral cells with histamine, there's essentially no effect on the stimulus-evoked responses of these cells. But now when we bath apply antagonists of inhibitory neurotransmitter receptors, we essentially abolish warming-evoked excitation in these cells, which again is very similar to the result that we saw in the GR28BD mutant, which is, I think, sort of modest evidence that that really is the only peripheral warm transduction molecule. So I think this is our conjectured circuit. When we silence these peripheral cells with histamine, there's still a secondary pathway for warming-evoked excitation, but that depends critically on intact inhibition. Finally, and I'll just zoom through this fourth cell type quickly, we found a type of projection neuron which is selectively excited by really rapid changes in temperature in either direction. So I'm calling these warm-cool projection neurons. At the onset of these rapid stimuli, you can see they fire a brief burst of spikes. You can see this in the peristimulus time histograms. And the story here is much the same as the story for the warm projection neurons. So these responses are not blocked by blocking inhibition. They're not blocked by the GR28BD mutant, even this warming-evoked excitation, which is a little surprising. But in the context of this genetic background, we'd expect these responses to be blocked by blocking inhibition, and that's indeed the case. So in the GR28BD mutant, warming-evoked excitation is now completely blocked by blocking inhibition. So we think then that this is the circuit diagram for the central representation of sort of really rapid temperature change. I think it's quite similar to the wiring diagram for the representation of warming. The only twist might be that this projection from the cool peripheral neurons may be sort of relatively stronger, such that these become warm-cool neurons rather than just warm responsive neurons. So in all these diagrams, I have shown you sort of a hypothetical inhibitory interneuron that's excited by cooling. Does that kind of interneuron exist? So to test that idea, we labeled GABAergic neurons genetically. We recorded from many of them randomly in this brain region. And I'll tell you that a strong majority of the cells that we randomly targeted were indeed excited by cooling and inhibited by warming. Importantly, they had pretty substantial rates of spontaneous firing, such that there's some dynamic range for disinhibition. And I'll show you that this is kind of even clearer when you look at the response to very small temperature steps and you look at both the memory potential change and the change in firing rate. So we think that these cool local neurons are potentially a good substrate for playing this role in the circuit and essentially inverting the sign of this cool pathway input. So in conclusion, our data suggests that there is an asymmetry in the central representations of warm versus cool in the Drosophila brain. So the cool projection neurons seem to be participating in circuits that are really feed-forward excitatory circuits. However, the neurons that are excited by warming, the warm projection neurons and the warm-cool projection neurons, these neurons seem to be driven by more complicated circuits that combine excitation from opponent neurons as well as inhibition driven by one of those opponent pathways. There's also an asymmetry, interestingly, in the number of neurons representing warm and cool. So what we found is that on each side of the brain, there appear to exist, as far as we can count, 6 cool projection neurons, 1 warm projection neuron, and 1 warm-cool projection neuron. The caveat here is that there's no guarantee that we've identified all the projection neurons in this brain region, but a fairly systematic and unbiased sampling gave these numbers. Interestingly, there's a similar asymmetry exists in the periphery. So in the antenna, there are more cool activated neurons than warm activated neurons. And I was quite interested to see in the mammalian literature, the exact same thing is true. In both the skin and in the CNS, in the spinal cord. So why combine warm inputs and inverted cool inputs? Why might warm projection neurons do this? I want to reinforce the point that Dora Angelaki made very eloquently, which is that pooling from more signal sources should in principle improve the signal-to-noise ratio of neural activity. Keep in mind that both the warm peripheral cells and the cool cells are informative about the existence of a warm stimulus. It's just that cool cells are speaking a different language. They're inhibited rather than being excited. But this is a perfectly valid source of information about peripheral thermal stimuli. So it makes sense that you might benefit from inverting this signal and then just pooling it with warm-evoked excitation. There's another argument as well. And again, this really follows upon a point Dora made about all neural circuits being noisy, and noise being a challenge when stimuli are ambiguous and weak and close to the noise. And the point I want to make here is that this circuit should cancel non-thermal noise that's positively correlated in warm and cool neurons. So this cartoon is supposed to represent the existence of, for example, a mechanical stimulus. These neurons reside way, way on the fringe of the body, right? And one might think that this is actually important for their function. If they're going to be sensitive sensors of peripheral temperature changes, they might need to have this rather exposed location. But this exposed location also opens them up to all kinds of mechanical perturbations. When the fly, for example, bumps into an object, if that bends the arista, all neurons are essentially non-specifically excited by bending. That would be expected to produce correlated activity in these peripheral neurons. And that's in contrast to their true sensory stimulus, right, which will produce anti-correlated activity in these neurons. And so what this circuit does is that it looks for specifically anti-correlated activity, and it passes only those signals that are anti-correlated. Stimuli that produce correlated activity are treated as noise and are essentially canceled out by this circuit. That's our speculation. Finally, if you'll suffer through one more speculative slide, why is hot not simply the opposite of cold? Why is there this asymmetry both in the topology of central circuits and in the numbers of neurons that are devoted to explicitly representing either warm versus cool stimuli? So one idea is that there may be a difference in the statistics of warm and cool stimuli in the natural world. For example, I know of no literature for or against this idea, but it should be easy to test. There may be different biophysical constraints on the molecules that transduce warming and cooling. We don't know much about these molecules yet. GR28BD is newly identified. The cold receptor is not yet identified in the fly. Finally, cool peripheral neurons may have evolved first. So interestingly, there are a number of insects, including ants and cockroaches, that do not have peripheral warm activated neurons. They have only peripheral cool activated neurons. But yet these insects still avoid warm temperatures. And in the brain of at least the cockroach, workers have identified central neurons that are specifically excited by warm stimuli. So the speculation has been that these neurons are driven by the kind of disinhibitory loop that I described to you that our evidence suggests. So it may be that that's actually the more ancient circuit motif, and that the evolution of peripheral warm sensors has sort of piggybacked on that circuit. So finally, I just want to draw attention to the really fabulous people who did this work. Wendy Liu was a student in my lab for the last several years. She's recently gone back to medical school. This project was entirely her own idea, and it was heroic. And she's now thinking hard about how to translate discoveries at the bench to benefiting patients. And then Ofer Mazur is the director of our really great research instrumentation core facility. He's responsible for designing and building the thermal stimulation device. Finally, thank you to all the people who really generously gave us flies or assistance and to our funding sources. Thank you.
Is it known how the peripheral neuron, the cool neuron, become hyperpolarized by heat? And is that also found in vertebrates?
It is clear, at least on my reading of the literature, that vertebrate peripheral thermosensory neurons are bidirectionally responsive. So cool preferring neurons are inhibited by warming, and warm preferring neurons are inhibited by cooling. How this works at the molecular level is not entirely clear, but I think it seems likely a priori that there's some basal level of transduction and that the probability of opening of the transduction channel is actually suppressed by its non-preferred stimulus. How warm and cool sensing occurs at the level of channel biophysics is still, I think, fairly controversial and interesting. Yeah.
So in your model, one prediction might be that if you got rid of the cool neurons, you would lose the GR28 beta the independent warming response in the projection neurons.
That's right.
Have you done that? Do you see that?
We've tried very hard to do that experiment. The problem is that we and everyone else in the field have been unable to identify a driver line which is both powerful and specific for the cool peripheral neurons. Several not very specific lines have been identified and published. We identified a line that we thought was pretty specific. We actually have to convert it into a LEX-A line to do this experiment, which I didn't tell you about for the warm version of that, but Wendy had to work hard on that. And our LEX-A version of that is quite weak. It drives only weak GFP expression. And in the positive control experiment, we record from the cool projection neurons. We don't see much effect of silencing the peripheral neurons with histamine. Again, consistent with it's really weak drivers. So we just need somewhat better reagents. If someone could discover the peripheral cool transduction molecule, that would help a lot. Yeah.
I have two questions. First one is about GR28B expression. Is it expressed in other appendages in the fly, and do those neurons also project to the same common region, or are there several centers for cool and warm?
Right. Interestingly, GR20BD is expressed in several peripheral structures. There's a really nice paper from Hubie Amrijn's lab that described that several years ago. It's expressed in the legs, I think, some places in the wing, and I think some places in the mouth parts. Whether those neurons feed into satellite thermosensory processing regions, for example, in the ventral nerve cord, is not clear. But I would place some money on the idea that there actually are there's probably more than one peripheral thermosensory organ in the fly.
My second question was about whether the second order projection neurons, then you described two projection areas, the protocerebrum and the mushroom body. Do any of these regions have neurons that respond to pain or that sort of are painful, that convey pain to the fly? So does the hot and cold actually, too much hot or too much cold, will that be painful to the fly like this?
There's some work that's been done on the peripheral sensing of sort of quite noxious stimuli in the fly. For example, Dan Tracy's lab has worked on a molecule that they call painless. But to my knowledge, there's little or nothing known about central representations of painful stimuli in the fly. Yeah.
Very interesting, very interesting, Rachel. So have I understood correctly that it is on-off? It's like a digital system? There is no analog coding at all in terms of different levels of temperature or different levels of rate of change of temperature? And if not, then what's the baseline?
Right. So these neurons are actually analog encoders. For simplicity, I showed you only data that was a warming of about 10 degrees or a cooling of about 10 degrees. But these neurons are actually pretty sensitive. So let's see. This is just an example of intensity response function for the two cool projection neurons, but I could show you similar kind of data for the warm responsive neurons. But I think these make an interesting contrast. So here I'm showing you the responses to really small temperature steps, ranging from 1/10 of a degree to 2 or 3 degrees. You can see that, for example, the highly adapting cool responsive neuron is also very sensitive. That probably makes some kind of sense. The less adapting cool neuron is comparatively less sensitive. But as a population, these neurons certainly are sensitive to very small temperature changes. So they are analog encoders rather than digital encoders.
So I have a question about the local microcircuit in the warm projecting neurons. It receives the projection from the warm primary neurons and the cool primary neurons, as well as the inhibitory projection from the cool projection neurons. And how does the blocking inhibition become dominant in that local microcircuit? And also, what is the meaning of the projection of the cool
Yes.
Neurons into the warm projection?
I kind of glossed over that, in part because I don't have a great story to tell about it. So the circuit that is being referred to here is the fact that in these warm preons, and we think also in the warm-cool preons, there's not only direct excitation from warm cells, disinhibition from cool cells in response to a warming stimulus, but also this kind of funny direct projection from the cool cells. We think it's weaker just because it's effectively masked by this disinhibitory projection, and the net effect of warming is to excite these cells. But it's quite curious. I don't have a great just so story. I would just point out to you that in most central neurons, most stimuli in the world that drive any type of activity in that neuron drive either concurrently an increase in excitation and inhibition or a decrease in excitation and inhibition. E and I inputs almost kind of always go together on average. And it's sort of small distinctions in the magnitude of timing of E and I that really control neural activity in general. So I would argue that it's perhaps not so surprising to see co-recruitment of excitation and inhibition onto this neuron in response to a cool stimulus, for example. And I'll just say one more really brief thing in response to this is that, I hope I don't flub this. There's some analogy to spinal circuits here. So there are nociceptive neurons in the spinal cord of mammals that receive input from nociceptive peripheral neurons. They also seem to receive excitation from mechanosensory afferents, so neurons in the periphery that respond to innocuous mechanical stimuli like the feeling of the clothes on your body. But that's normally masked by mechanosensory-driven inhibition by local interneurons. I'm drawing here the conjectured circuit that I think the field kind of agrees on. And then in pathological conditions, often this inhibitory input is removed such that you get a net excitation by mechanosensory afferents. And it's thought that this sort of circuit motif is what gives rise to mechanical allodynia, the perception in pathological states that even the feeling of your clothes on your body is excruciatingly painful. The normal function of the circuit is not entirely understood, but the idea that there's this funny input which is normally masked by co-recruited inhibition is kind of the analogy I'm trying to draw with our findings. And I wonder whether looking at kind of relatively simple circuits like the one I've described might provide some insight into circuits like this where a kind of a strange off-target stimulus is normally masked by co-recruited inhibition.
Thank you, Rachel. In the interest of time,